Hello everyone, my name's Iman. Welcome back to my YouTube channel. Today we're going to cover Chapter 8 in our MCAT Biochemistry series, and this chapter is titled Biological Membranes. In this chapter, we're going to cover the following objectives. First, we're going to discuss the fluid mosaic model, and this is going to include the general structure and function of membranes, as well as how membranes remain dynamic in their composition and movement. Then we're going to look closely at membrane components, including the different types of lipids, proteins, and carbohydrates that make up the membrane. We're also going to talk about membrane receptors and the cell-to-cell junctions that will allow communication and connection between cells. Next, we'll explore membrane transport, which covers how substances move across membranes through concentration gradients, passive transport processes, and the active processes of endo and exocytosis. Finally, we're going to look at specialized membranes, and we're going to focus on how cells maintain a membrane potential and how mitochondrial membranes are adapted for energy production. With this introduction, let's go ahead and get started with Objective 1 and discuss the fluid mosaic model. The cell membrane, which is also called the plasma membrane, it's often described as a semi-permeable phospholipid bilayer. Semi-permeable means that it's going to allow certain substances to pass through while restricting other substances. And this property is very important for maintaining the internal environment of the cell. The fluid mosaic model is the theory that explains the structure and function of this membrane. And it tells us that the membrane is not a rigid structure. Instead, it is flexible with lipids and proteins moving within it, almost like a dynamic, constantly shifting landscape. With that being said, the cell membrane has three main jobs. First, it protects the cell's interior from the external environment. Second, it's going to regulate the exchange of materials. It's going to make sure that essential nutrients can enter, that waste products can leave, and that harmful substances are kept out of the cell. Third, it plays a role in communication and signaling, and that's going to allow the cell to respond to its surrounding. The structure of the bilayer is very key to how all of this works. Phospholipids are arranged so that their hydrophobic fatty acid tails are pointed inward away from the water, and their hydrophilic phosphate heads, they are going to face outwards towards the aqueous environments inside and outside of the cell. This arrangement creates a stable barrier that still allows movement. Now, phospholipids, they can move. They can move laterally within the same layer of the membrane, and this gives the membrane its fluidity. This lateral movement is the fastest type of molecular motion in the membrane, and it happens frequently. This is in contrast to a different type of movement called flip-flop, which is very slow. So the movement of phospholipids between layers, that's called flip-flop, and again, it's very slow without the help of enzymes. Proteins within the membrane, they can also move laterally, but their movement is going to be generally slower because of their larger size and also because of the interactions they have with other cell structures. On that note, the membrane is more than just phospholipids. There's proteins, there's carbohydrates, there's a lot to discuss. As for proteins, they're going to be embedded in or attached to the membrane, and they serve important functions. Some proteins are going to transport molecules in and out of the cell. Others are going to receive signals from the environment. And some are going to provide structural support by anchoring to the cytoskeleton or to the extracellular matrix. Carbohydrate chains, those are often attached to proteins or lipids on the extracellular side, and they form things like glycoproteins and glycolipids. These carbohydrate groups, they're going to be critical for things like cell recognition, for cell communication, and even for forming protective coats. We'll also see areas called lipid rafts. These are specialized microdomains in the membrane, and these regions are going to be rich in cholesterol and sphingolipids, and they play a role in organizing signaling molecules. Because Lipid rafts are more ordered and they're tightly packed in comparison to the surrounding membrane. They can act as platforms for certain cellular processes. Another important feature of membrane organization is asymmetry. The composition of phospholipids in the inner and outer layers of the bilayer, they're not the same. And this asymmetry is actively maintained by enzymes called lipase. These are going to help move specific phospholipids from one leaflet of the bilayer to the other when needed. Finally, we have to note that the membrane is adaptable. Its lipid and protein composition, as well as the distribution of receptors, those can change depending on the cell's needs and environmental conditions. And this ability to adjust is going to be important for the cell's survival, for signaling, and just for overall function. With that, we can move into objective 2, which is all about membrane components. A lot of the content in this objective we've already touched on in chapter 5, but it's worth revisiting them in detail and then tying everything together. Biological membranes are made up of a variety of molecules, and each of these molecules are going to contribute to structure, stability, and function. We're going to begin with the lipids. Fatty acids and triacylglycerols form a very good starting point because they set the stage for the more complex membrane lipids we're going to encounter. A fatty acid is going to have a long hydrophobic hydrocarbon chain that repels water and a hydrophilic carboxylic acid group that can interact with it. This amphipathic nature affects how these molecules organize in water and how they interact with other cellular components. Triacylglycerols, also called triglycerides, they are storage lipids rather than structural ones, but their chemistry is important to understand. They consist of 3 fatty acids linked to a glycerol backbone by ester bonds, and they're formed through a condensation reaction that releases water. Further, fatty acids, they can be fully saturated. This means they will contain only single bonds and they're going to pack tightly to form solids at room temperature. On the other hand, they can also be unsaturated with one or more double bonds that will introduce kinks. In this case, that will prevent tight packing and it will actually make them liquid at room temperature. Something important to note here is that most naturally occurring unsaturated fatty acids have cis double bonds, which will create noticeable bends in the hydrocarbon chain. This feature increases membrane fluidity. Trans double bonds, however, they keep the chain relatively straight, and they're going to allow tight packing similar to saturated fatty acids. That means they're going to reduce membrane fluidity. And this structural difference really plays a key role in how different lipids influence the physical properties of biological membranes. From here, we can move into phospholipids. One major subtype is the glycerophospholipids, also called phosphoglycerides. These molecules are going to have a glycerol backbone with 2 fatty acids attached by ester linkages and a phosphate group that's attached by phosphodiester linkage to a highly polar head group. The type of head group that can vary, and this variation determines the molecule's surface properties. In addition, the chemical nature of the head group, whether it's neutral or positively charged or negatively charged, that's going to influence how the phospholipid interacts with proteins, how it is going to participate in signaling, and how it contributes to the membrane's overall structural properties. Phospholipids are also amphipathic, meaning they have both hydrophobic tails and hydrophilic heads, which is going to allow them to spontaneously assemble into micelles or liposomes in aqueous environments. Now, not all membrane lipids are built on glycerol. Sphingolipids, for example, use a sphingosine backbone instead. They still have a long hydrophobic tail and polar head groups, but their chemistry and their biological roles, they can be quite different. Sphingolipids include several subtypes such as ceramide, sphingomyelin, glycosphingolipids, and gangliosides. And each of these molecules has specific functions. But overall, we can say that sphingolipids, they play big roles in both membrane structure and cell signaling. As a friendly reminder, ceramide is the simplest sphingolipid. It's going to serve as the building block for the others. We really highlighted this in chapter 5. Now, when a phosphocholine group is added to ceramide, it's going to form sphingomyelin. When one or more sugar molecules like glucose or galactose are attached, you're going to get cerebrosides. If additional sugars and sialic acid are added, then the molecule becomes a ganglioside. And these modifications, they're going to happen in a stepwise manner. So sphingolipids are sequentially built to form the biologically important molecules in this class. Now, because of their structural diversity, sphingolipids, again, contribute to things like membrane stability. They contribute and participate in cell recognition, and they can act as signaling molecules in various pathways. Now let's talk about steroids, and then we're going to focus in on cholesterol. Steroids are a class of lipids that all share the same basic core structure. That structure is made up of 3 cyclohexane rings and 1 cyclopentane ring fused together. Even though the base structure is the same, the oxidation state and the different functional groups that attach to it can vary, and those variations are what give each steroid its unique properties. One important group of steroids are the steroid hormones like cortisol, estrogen, and testosterone. These hormones are going to bind to high affinity receptors, which means they can trigger big effects even at very low concentrations. And they're also involved in processes like regulating metabolism, controlling inflammation, maintaining salt and water balance, and even influencing gene expression. We really go over the effects of steroid hormones in our biology playlist, so hopefully you're checking that out as well. Now let's shift to cholesterol. Cholesterol is also a steroid. It's the starting point for making every other steroid in the body. Without cholesterol, you cannot synthesize steroid hormones. But cholesterol's role goes beyond being just a precursor. It's also a key structural component of cell membranes. Because of its rigid ring structure, cholesterol helps maintain the shape and the stability of the membrane while also playing an important role in regulating membrane fluidity. So at higher temperatures, cholesterol limits how much the phospholipids can move around. So that's going to prevent the membrane from becoming too fluid or too permeable. At lower temperatures, it's going to disrupt the tight packing of the phospholipid tails, which prevents the membrane from becoming too rigid or too solid. In other words, cholesterol kind of acts like a buffer that keeps membrane fluidity in check when the temperature changes. And to top this all off, cholesterol is also the precursor for bile acids, which are needed for digesting fats, and also for vitamin D, which is important for calcium balance and bone health. Finally, the last lipid we're going to cover is waxes. Waxes are esters made from long-chain fatty acids linked to long-chain alcohols. They are extremely hydrophobic, which makes them great at repelling water. In plants, waxes are often found as a coating on leaves and fruits, and this coating really helps reduce water loss and protect against microorganisms. In animals, waxes appear in places like earwax, where they act as a barrier and they can have antimicrobial properties. In short, what we need to know is that most waxes serve an extracellular function in protection or waterproofing. Besides lipids, proteins are also major components of biological membranes, and they play a lot of roles in membrane function. The fluid mosaic model accounts for three main types of membrane proteins we need to know for the MCAT. The first type is transmembrane proteins. These proteins span the entire lipid bilayer, meaning part of the protein is exposed to the extracellular environment, and then part of it is going to be exposed to the intracellular environment. They often function as transporters, channels, or receptors, and because they cross the whole membrane, they can directly communicate signals or even move substances between the inside and the outside of the cell. The second type is embedded proteins. These proteins are associated with only one side of the lipid bilayer. So depending on their orientation, they can face either the cytoplasmic side or the extracellular side. And even though they don't span the whole membrane, like transmembrane proteins, embedded proteins are still involved in processes like signaling or anchoring structures to the membrane. The third type is peripheral proteins. These do not insert into the lipid bilayer. Instead, they kind of attach loosely to the membrane surface, either through things like electrostatic interactions with the lipid head groups or by binding to other membrane proteins. Some peripheral proteins associate with lipid rafts, which again are small, specialized microdomains in the membrane that are enriched in cholesterol, sphingolipids, and specific proteins. And because lipid rafts are more ordered than the surrounding membrane, they can serve as platforms for certain processes. Peripheral proteins that localize to these rafts, they're often going to participate in signaling, or they're going to help maintain the cell's shape by linking to the cytoskeleton. This association is especially important in signaling because lipid rafts can bring together the receptors, enzymes, and other components of a pathway in one place, and that will make communication inside the cell faster and more efficient. Now, the term integral proteins, this is used to describe both transmembrane and embedded proteins. These proteins interact closely with the hydrophobic interior of the membrane, and they often require detergents or other special treatments to be removed from it. Because of their positioning, integral proteins are critical for functions like transport, for signal transduction, and of course, structural support. On that note, let's also talk about membrane receptors, which are often transmembrane proteins. These receptors are going to detect specific signaling molecules outside the cell, and then they're going to trigger responses inside the cell. Many receptors, especially enzyme-linked receptors like receptor tyrosine kinases, they are transmembrane proteins with catalytic activity. So when a ligand binds to the extracellular portion, the intracellular portion can act as an enzyme, often like adding phosphate groups to target proteins to then initiate the signaling response. Other receptors, they might directly activate or deactivate transport proteins, and that can control the movement of ions or molecules across the membrane. Still, others are going to initiate a cascade of intracellular signaling events that can go on to either alter gene expression, have some role in enzyme activity, or even cellular metabolism. So, for example, ligand-gated ion channels, they open when a specific molecule binds, and they're going to allow ions to flow into or out of the cell. Others, like G-protein coupled receptors, they can go ahead and initiate complex signaling pathways. In addition to lipids and proteins, carbohydrates are also going to be important components in biological membranes. And these carbohydrates are usually going to be found attached to proteins where they can form glycoproteins, or attached to lipids where they can form glycolipids. They are almost always going to be located on the extracellular surface of the membrane, and the hydrophilic nature of these carbohydrate chains is going to allow them to not only attract, but hold water at the surface. This creates a protective and hydrated layer around the cell, which will shield the membrane from mechanical and chemical damage. And also, it's going to help maintain the cell's local environment by trapping certain molecules close to the surface. Carbohydrates are also essential for cell recognition, so they can act like identification tags that can be read by other cells or by molecules in the extracellular space. The immune system, it relies on those tags to distinguish between self and non-self cells. This is why certain pathogens, like viruses and bacteria, they target membrane carbohydrates to gain entry into cells or to identify a host cell that they can actually infect. And blood type antigens, those are a well-known example of membrane carbohydrates that determine immune compatibility. With that in mind, what we want to move to next is cell-to-cell junctions. Cells, they build cohesive layers using junctions. And these junctions, they either 1, seal spaces between cells, 2, anchor the cells so the tissue can resist stress, or 3, create direct channels for communication. They can also connect cells to the extracellular matrix, like the basement membrane you're going to see under epithelium. Let's begin with tight junctions. Tight junctions sit near the apical surface of epithelial cells, and they work like a zipper that seals the space between adjacent membranes. By sealing the paracellular route, tight junctions can then control what can slip between cells. They also preserve cell polarity by preventing lipids and membrane proteins from diffusing between surfaces. This is going to keep transporters and receptors in the right place. Next are desmosomes. These act like spot welds. They use cadherin family proteins to link neighboring cells. And inside the cell, these cadherins connect to intermediate filaments like keratin through dense protein plaques. The result is mechanical strength. So tissues that experience a lot of shear, like our skin and cardiac muscle, they rely on this so that cells do not tear apart when the tissue is stretched. Closely related to this are hemidesmosomes. These anchor a cell to the basement membrane using integrins, and inside the cell, they also attach to intermediate filaments. So you can think of desmosomes as cell-to-cell anchors and hemidesmosomes as cell-to-matrix anchors. Both are going to protect tissues from mechanical stress. Now, we'll talk about gap junctions. Gap junctions create direct cytoplasmic channels between neighboring cells. Each channel is built from 2 connexons, and each one is made of 6 connexin subunits. These pores allow ions and small molecules to pass from cell to cell, which really helps synchronize activity. Cardiac and some smooth muscle depend on gap junctions so that electrical signals can spread quickly and contractions can happen in a very coordinated way. But important to note that gap junctions are going to be sensitive to conditions like low pH or high calcium, which can close the channels to protect cells during injury. Now, a quick note here, in animals, the main cell-to-cell junctions are tight junction, desmosomes, and gap junctions. Plasmodesmata, those are similar to gap junctions in function, but they're found only in plants, where they kind of cross the cell wall to link the cytoplasm of adjacent plant cells. We don't need to know any more details about this, but all of these junctions, tight junctions, gap junctions, and desmosomes are supported and organized by proteins called cell adhesion molecules, or CAMs. One of the most important role of CAMs is to help cells recognize one another. And this recognition allows cells to connect in the right way, which is important when tissues are forming. So CAMs can also guide cells to their correct positions during growth and repair. In addition, they influence how cells mature into their specialized roles. This is a process known as differentiation. By regulating how cells attach and communicate then, CAMs have a very major influence on how tissues develop and function over time. With that, we have completed objective 2, and we can move into objective 3. This section is all about how substances move across cell membranes, and that movement, it can happen in different ways depending on whether or not the cell needs to use energy. Transport processes fall into 2 broad categories: passive transport and active transport. Passive transport doesn't require any energy input from the cell. Instead, it just relies on the natural movement of molecules from an area where they are more concentrated to an area where they are less concentrated. And this movement is driven by something called a concentration gradient. In contrast, active transport is going to require the cell to use energy, usually in the form of ATP, to move substances against their concentration gradient. So from an area of low concentration to an area of high concentration. But let's start with passive transport first. There are 3 main types: simple diffusion, osmosis, and facilitated diffusion. Simple diffusion is the movement of small nonpolar molecules directly through the lipid bilayer without any help from proteins. Oxygen and carbon dioxide are good examples because they're small and nonpolar, so they can slip easily between the lipid tails. Now, some substances, they can't cross the membrane on their own, and they need a little bit of help. This is where facilitated diffusion comes in. Please note, it's still passive. It does not require energy, but it does rely on specific proteins in the membrane to help molecules move across. And these proteins, they can be channels which form pores that allow molecules or ions to flow through, or they can be carriers which bind to the molecule, change shape, and then release it on the other side. Large polar molecules like glucose and ions like sodium and potassium, they often use this type of transport. Another type of passive transport is osmosis. This is the movement of water across a semi-permeable membrane. In osmosis, water moves from the side with a lower solute concentration to the side with a higher solute concentration. And it works to balance the concentration on both sides of the membrane. This movement is driven by differences in solute concentration, so it does not require energy. The effect of osmosis on a cell depends on the tonicity of the surrounding solution. So in a hypertonic solution, the surrounding fluid is going to have a higher solute concentration than the cell interior. And because we know water moves towards the higher solute concentration, water will leave the cell and it's going to cause the cell to shrink. But in a hypotonic solution, the surrounding fluid actually has a lower solute concentration than the cell interior. So water is going to enter the cell and it's going to move towards the higher solute concentration inside. That's going to make the cell swell and potentially burst. In an isotonic solution, the solute concentration is going to be equal inside and outside the cell, so water moves in and out at equal rates, and the cell maintains its normal size. From here, we can introduce osmotic pressure. Osmotic pressure quantifies the force that drives water movement during osmosis. Osmotic pressure is a colligative property, so that means it depends only on the number of dissolved particles in a solution, not their identity. Whether the solute is a salt, a sugar, or any other molecule, osmotic pressure depends solely on how many particles are present. The osmotic pressure of a solution, it can be calculated using the equation you see here on the page. This is osmotic pressure. I is the Van Hoff factor, which is the number of particles a solute dissociates into. We go into a lot more details on this equation in the Gen Chem MCAT playlist if you want to touch up on that. But again, I is the Van Hoff factor, M is molarity R is the gas constant, and T is the absolute temperature in Kelvin. And this equation shows that osmotic pressure increases with the number of dissolved particles, their concentration, and the temperature. In living cells, I want to note that osmotic pressure is not acting against gravity, but rather against the cell membrane. So this means cells have to maintain a careful balance between solute concentrations inside and outside to prevent excessive water gain or loss. And that is a process that is super essential for maintaining cell structure and function. Now, in contrast to passive transport, active transport moves solutes against their concentration gradient, meaning we're going from an area of low concentration to an area of higher concentration. Because this movement works against the natural flow of diffusion, it's going to require input of energy. And this energy can come directly from ATP or indirectly from the energy stored in another gradient. Primary active transport uses ATP directly to power the transport process. So here, specialized proteins in the membrane, often transmembrane enzymes called ATPases, they're going to hydrolyze ATP into ADP and inorganic phosphate, and in that process, they will release energy. This energy is then used to change the shape of the transporter protein and then move molecules across the membrane. An important example is the sodium-potassium pump, which uses ATP to move sodium ions out of the cell membrane and potassium ions in, both against their concentration gradients. Now, secondary active transport, it doesn't use ATP directly. Instead, it uses the energy that's stored in an existing electrochemical gradient created by primary active transport. So in the system, the movement of one particle down its gradient powers the movement of another particle against its gradient. If both particles move in the same direction across the membrane, this is called symport. If they move in opposite directions, it's called antiport. For example, the sodium glucose symporter uses the sodium gradient to drive glucose uptake into cells. In addition to active transport through pumps and carriers, cells also move large volumes of material through bulk transport processes like endocytosis and exocytosis, which rely on vesicle formation and fusion with the cell membrane. Endocytosis is going to occur when the cell membrane folds inward and engulfs material to bring into the cell inside a vesicle. And this can take different forms. We can have pinocytosis, which is the uptake of fluids and dissolved substances. And we can have phagocytosis, which is the engulfing of large particles or solids, things like cellular debris or microbes. Exocytosis is the reverse process. Here, vesicles formed inside the cell fuse with the cell membrane and then release their contents into the extracellular environment. And this is how cells secrete hormones, neurotransmitters, and other important substances. With that, we can move into our last and final objective titled specialized membranes. Now, while all cell membranes share the same general structure, a phospholipid bilayer with embedded proteins, some membranes are adapted to carry out very specific functions. One important specialization that we need to talk about is the ability of a membrane to maintain a membrane potential. A membrane potential is the voltage difference across the membrane, and it's created by the uneven distribution of ions between the inside and the outside of the cell. This difference in charge, it's not just a byproduct, it's an essential feature of cell physiology. And maintaining a membrane potential, that's going to require energy because over time, ions tend to passively diffuse across the membrane through leak channels, and they're going to move down their concentration gradients. To then counter this, Cells use ion transporters or pumps, things like sodium-potassium pump, for example, to actively regulate ion concentrations. Then to quantify membrane potential, we can use the Nernst equation, which allows us to calculate the equilibrium potential for a given ion based on its concentration inside and outside the cell. This equation takes the gas constant, the temperature, the ion's charge, and the ratio of outside to inside concentrations into account. And the form we often use in biology simplifies to 61.5 divided by the ion's charge, multiplied by the log of the concentration outside over the concentration inside. This tells us the electrical potential at which there is no net movement of that ion. However, in real cells, the membrane potential is going to be influenced by multiple ions at the same time. That's where the Goldman-Hodgkin-Katz voltage equation comes in. This equation is going to account for the permeability of the membrane to each major ion, commonly sodium, potassium, and chloride, and all their relative contributions to the overall membrane potential. If, for example, the membrane is more permeable to potassium than sodium, potassium is going to have a greater influence on the resting membrane potential. Now, one of the key players in maintaining this potential is the sodium-potassium pump, or sodium-potassium ATPase. This pump uses ATP to move 3 sodium ions out of the cell and 2 potassium ions into the cell with each cycle. This is not only going to keep intracellular sodium low and intracellular potassium high, but it also contributes to the cell's negative resting potential. Because sodium and potassium carry positive charges, this pumping action is electrogenic. It creates a net movement of positive charge out of the cell, which helps maintain that voltage difference. Now, this is a process we discuss heavily in Chapter 4 of our MCAT biology playlist if you want to check that out for more information. But from here, I want to note that specialized membranes, they're also critical in organelles. Take the mitochondrial membrane as an example. The outer mitochondrial membrane, it's relatively permeable to small molecules and ions because it contains large pores. But in contrast to that, the inner mitochondrial membrane, it's highly selective. It's impermeable to most ions and molecules unless specific transporters are present. It's also folded into structures called cristae, which greatly increase the surface area that's available for the electron transport chain and ATP synthase. Another important point is that the inner membrane, it lacks cholesterol, unlike many other cellular membranes. So together, all these features allow the mitochondria to efficiently produce ATP, with the inner membrane enclosing the mitochondrial matrix where those key metabolic reactions occur. With that, we've completed this chapter. I truly hope it helps. Please let me know if you have any questions, comments, concerns down below. Other than that, good luck, happy studying, and have a beautiful, beautiful day, future doctors.